Good morning. I am Dr. Madhuramozhigovindarajulu, Head and Associate Professor in Zoology, A.D.M. College for Women, NGAPATTINAM. Today, I am going to present the topic of applications of biotechnology in medicine. And coming to biotechnology, this is nothing but manipulation of the gene. We are going to manipulating the gene through genetic engineering, and in what way we can help this technology in the medical field. So coming to the detecting and diagnosis of human diseases, we can use this biotechnology as molecular diagnostics, because the use of DNA, RNA and proteins to facilitate disease detection, diagnosis, subclassifications, prognosis and monitoring response to therapy. What do you mean by therapy? Therapy is nothing but the treatment. Here we are using the gene therapy for various diseases diagnosis and also for curing disease also. So coming to the advantages of the molecular diagnostics, this is mainly because improvement in the sensitivity. We can use through the biotechnology, the improvement in sensitivity, high specificity, because each gene is controlling the DNA, and this gene, that is the DNA, which is controlling the enzyme production. In turn, they are very important for the protein synthesis. So one gene, one enzyme hypothesis is applied because they are high specificity, and also this treatment, this therapy is costless, and also for faster analysis of the timing, because they will take a very faster time to analyze the disease. So for the diagnostic purposes of various diseases, we can go for the biotechnology. So detecting the genetic disease. What is genetic disease? In some karyotype, we can see the change in the gene due to mutation. Similarly, the COVID treatment also shows the mutation of the gene. So fetal testing for chromosome abnormality and for the defective gene. What is chromosome abnormality? Usually we will find the genes in a linear manner inside the chromosomes. The chromosomes are arranged in the nucleus, isn't it? But here the chromosomal abnormality occurs due to various cancer gene or anything, the carcinogen, which are responsible for the mutation, and because of the mutation they cause chromosomal abnormality, that is the addition or deletion of the gene in the chromosome, duplication, inversion. Likewise, any abnormality can occur. So this causes mutation. So fetal testing means we are testing the fetus for the genetic diseases. So taking the amniocentesis, this is the test. We are taking the amniotic fluid. What is amniotic fluid? The embryo which are floating in the amniotic fluid of the mother's womb. So we can take this fluid and testing at 16 weeks of the karyotype, that is 16 weeks of pregnancy, we can identify if there is any abnormality of the embryo in future. Then chorionic villus sampling. This is also the test of a pregnant lady, that is the embryo at the stage of 8 to 10 weeks. We are taking the chorionic villi. What is chorionic villi? This is nothing but the chorionic membrane having intrusion into the, that is chorionic membrane is in the pregnant lady's uterus. This will be intruded into the embryo, fetus membrane, and they form a villus like structure, that is similar to the turkey towel structures. They are intruded into the fetus. So we are taking the sample from chorionic villi for testing any abnormality in the embryo. Then testing for chromosome abnormality and a defective gene. If a particular gene is defective, that means they cannot produce the enzyme In turn, they cannot synthesize the protein. So we can take the chromosomal abnormality test also through the genetic engineering and the biotechnology. Then fluorescence in situ hybridization, then fluorescence probe that is specific for chromosome and genes, that is the spectral karyotype. This can be also taken for the genetic diseases. Then dot plot methods. This is the genetic detecting the disease through the assay for detecting single nucleotide polymorphism. So using the PCR amplified DNA, what is PCR? This is the polymerase chain reaction. That means we can produce more number of the DNA by amplification. Amplification is nothing but multiplication and replication. So uses of PCR amplified DNA blotted onto the membrane. Then we can take the unbound ASO particles probe and this can be washed up, and bound ASO probe is detected by radioactive or calorimetric assay method. We can take the genetic diseases. We can diagnose the diseases. Next. And detecting the genetic disease, that is the single nucleotide polymorphism. Usually the nucleotides are arranged in the DNA, and the Watson and Crick structural model of DNA will show the double helical structure. We can see the sugar and the phosphate as a backbone. Then the nucleotides are connected in each strand from 3' to 1' or 5' direction. Likewise, it is the structural combination. So one of the most common form of genetic variation is the nucleotide polymorphism. So he estimated that one single nucleotide polymorphism occurs approximately every 1000 to 3000 base pair in the human genome. So when you take a human genome, 1 in 1000 to 3000 base pair. What is base pair? Base pair is nothing but the nucleotide. So there may be the polymorphism occurs and this can be detected by the biotechnology method. So nearly 99.9% of the DNA sequence will be exactly the same, and out of this only very rare nucleotide will cause the single nucleotide polymorphism. This is called the SNPs. So most have no effect because they occur in non-protein coding regions, that is they are introns. What is intron? There is because every gene, every nucleotide which are responsible for the production of a protein, but very few protein, very few gene are the nucleotide. They are not at all coding the protein. So these are called single nucleotide polymorphism. So most of them are intron, only very less amount of the single SNP occurs, approximately 1000 to 3000 base pair. So 10 pharmaceuticals are donated millions in a collaborative partnership called the SNP consortium. There are many pharmaceutical companies, they donated millions in a collaborative partnership, and this is they are making the SNP consortium. So coming to the chip microarray, this is nothing but the chip having thousands of pieces of a single stranded DNA molecule. Usually when you take a DNA, this is in a double stranded structure, but we are making a chip having single stranded DNA molecule, and this is isolated from a patient, fluorescently labeled and hybridized to microarray. So a laser scanner measures the intensity of the fluorescence to indicate the binding of the patient DNA to the single nucleotide polymorphism or gene on the microarray. So the chip is similar to the electronic device. This is having various number of the single stranded DNA molecules. So coming to the microarray, identifying the sets of diseases, gene diseases by microarray analysis. We can identify the disease. For example, the microarray created with known diseased gene or SNP and DNA from a patient is tagged with the fluorescent dye and then hybridized to the chip. So, binding of the patient DNA to a gene sequence on the chip, this indicates that the person's DNA has a particular mutation or the single nucleotide polymorphism. That is, due to the mutation occurs, there won't be the hybridization. So, we are taking the binding of patient DNA to a gene sequence on the chip and this indicates the person's DNA has a particular mutation or the SNP because the mutation occurs in the particular point. So, this is called the point mutation and this can be identified by means of microarray analysis. And then we can make various drugs and medicines through biotechnology. The main one is the RT-PCR that previous years most of the people they faced the COVID at the time. This RT-PCR is used to detect the COVID presence. The search for new medicine and drugs are going on nowadays mainly for the finding of oncogenes. What is oncogene? The genes which are responsible for the production of cancerous growth. So, the genes produce protein that may function as transcription factor and receptor for hormones and growth factor as well as serve as enzyme which involved in a wide variety of ways to change the growth property of a cell that cause cancer. Because oncogenes are the cancer producing genes, then tumor suppressor genes can be developed through the regulate oncogene. The gene regulation occurs, so we can use this gene therapy for the suppression of the tumor. Then personalized medicines. Then for breast cancer, BRCA1 or BRCA2, this can be used to increase the risk of developing breast cancer. Many other cases of breast cancer, they do not exhibit this mode of inheritance. So, they should be treated differently, that is the different chemotherapy. So, chemotherapy means this is nothing but the treatment. Radiotherapy means passing the sufficient amount of radiation to eradicate the cancerous or to arrest the or suppress the growth of the cancerous cells. Chemotherapy means using the treatment by chemicals. These chemicals can treat the cancerous cells growth, so they may suppress the cells. Then by means of nanotechnology. Nanotechnology is the area of science involved in designing, building and manipulating the structures at the nanometer level, very micron, that is nanomicron level. We can make the nanobots. This is manufactured device with embedded sensors to facilitate the target identification and drug delivery. That is the polymer based nanomedicines are used nowadays for various diseases. The production of vaccines and the therapeutic antibodies. Antibodies are produced in order to resist the disease, particularly the monoclonal antibodies, the vaccines which are stimulating the immune response. So, whenever our body is not having proper immune system, all disease will affect us. So, vaccine will stimulate our immune response. So, vaccination may be useful against conditions such as Alzheimer's disease or drug addiction. Using the antibodies in some type of therapies, the development of monoclonal antibodies are very important. The Alzheimer's disease mostly for the old age people and those who are addicted by drug and drinks, they can also get the vaccination of for relieving from the disease. Then gene therapy. Gene therapy is nothing but the treatment of the diseased gene or the defective gene. If we know the particular gene is diseased or defective, we can take the gene, or we can manipulate the gene, or we can alter the gene. So this is called the gene therapy. The gene therapy in various stages we can do, that is prenatal, postnatal, antenatal stages, we can do the gene therapy. So this is a vector delivers the therapeutic gene into a patient target cell. What is a vector? Vector is nothing but a plasmid. We cannot as such introduce a gene into your body. So we are taking the normal gene, or this is called as the desired gene, and this is incorporated with that of the plasmid, or this is a vector. Then we are culturing them, and we are taking them to the medium of bacteria for production of more number, and then this is introduced into a person who is having the defective gene. So functional proteins are created from the therapeutic gene, because the purpose of introducing the gene is to produce a protein, because if a gene is defective, they cannot produce a protein. That protein is absent means the metabolism will be affected. So we are introducing the normal gene or the desired gene instead of the defective gene, and this will produce a functional protein. So in the gene therapy, this was during their 1990, 4-year-old Ashanti Da Silva had a genetic disorder called severe combined immunodeficiency. This is nothing but the defect in the ADA gene, which results in an accumulation of the DATP, that is ATP is adenine triphosphate, DATP which will be converted into ATP, but if it is accumulated in a large quantity, this is toxic to the patient. So which is toxic to certain type of T cell. So this is takes down the entire immune system, because the immune system is similar to our circulatory system and going to various parts of our body. This DTP will be carried by the T cells to all our immune system. So our immune system will be affected. So the cystic fibrosis disease, this is also treated by means of a gene therapy. What is cystic fibrosis? So in our system, that is in the fibroid will be developing in the uterus or in the ovarian wall or any part of the body, and this can be treated by the gene therapy. So defective cystic fibrosis transmembrane conductance regulators are produced. Normally, it serves as a pump at the cell membrane to move electrically charged chloride atoms out of the cell. So if cells cannot move chloride out, they absorb water, trying to dilute the chloride in the cell, which leads to the production of a sticky mucus in our body. So this cystic fibrosis can be treated or remedied by the gene therapy. And what are the challenges of the gene therapy? That is definitely there may be the possibility of the adverse effects. So adverse effects of viral vectors, targeting the specific cells, controlling the expression of the therapeutic gene, then long lasting therapy. Some may require continuous long lasting therapy up to their death, and controlling the expression of the therapeutic gene. These are some of the, because we cannot tell all the genes that we are doing by gene therapy can be expressed. So for what purpose we are going for the gene therapy? Mainly for the expression of the gene, isn't it? So if you are transferring the gene or giving the treatment of the gene, sometime there may be the controlling the expression of the therapeutic gene. So these are some of the challenges we can face at the time of gene therapy. Then regenerative medicine, the growing cells and tissues, they can be used to replace or repair the defective tissues and organ. So tissue engineering, what is a tissue? So many cells they congregate to produce a tissue. Many tissues form the organ. So the tissue engineering is nothing but the genetic engineering, but the replacement of a tissue and organ by growing them in a culture. That means we are removing the organ which is completely destroyed. So instead of that, we are culturing the new tissue which are in a good condition. And then the cell-based treatment, then scaffold-guided tissue regeneration. This create framework onto which the cells are seeded and bathed in growth factor. In a growing factor, we are culturing the cells, and the cells can be seeded into the patient body. This is a regenerative medicine. Then cells-based tissue regeneration, fetal tissue grafting. This also done, that is the tissue from the fetus, because they are totipotent. They can develop into any type of cell. We can take them, and the nanofibers is also used for the regenerative medicine. And the bone regeneration, the blood vessels regeneration, they are having various experiments in this form, because the stem cells, they are having the high potentiality to develop into the normal and functional cells. So the transplantation of the bone marrow, this is also doing as an important advanced feature of the medicine. So cells and tissue transplantation, organ transplantation, these are very important process in biotechnology. And autografts, they are the transplanting a patient's own tissue from one region to body to another region. That is the vein from leg used in coronary bypass organ transplantation, or between the individuals also we can transplant the tissue, and so must be checked for the compatibility, that is the acceptability, because if there is any sort of rejection, this experiment, this surgery cannot be completed. So for autograft means taking the tissue from the same person. The organ may be different. That is, vein from the leg is used for the heart bypass, coronary bypass. So this is also happening. Then histocompatibility complex, that is 70 genes which produce tissue typing protein, and there are many different type of MHCs. They are proteins, one group called the human leukocyte antigens. So HLA, human leukocyte antigens, they have been using the immunosuppressive drugs, but there are some problems occur. But even though the HLA cells, that is the human leukocytes. So what is leukocyte? This is nothing but the white blood cell. So leukocytes are very, very important for our body. If any wound occurs, the leukocytes will come to the area where the wound occurs, and they produce the cells which are engulfing the drug, that is the antibodies or the bacteria from the external sources. So the leukoptosis occurs in the normal cases, and here the human leukocyte antigens, they are using for the suppression of the diseases, that is immunosuppressive drugs. They were used and a group of cells, they are called the human leukocyte antigens. They are used for organ transplantation and for tissue transplantation of the individuals. Cells and tissue transplantation, then organ transplantation, that is xenotransplantation, the transfer between the species, even from pig to human, the organ can be transferred. The University of Missouri scientists have produced cloned knockout pigs that lack the gene called GGTA1, that is the galactosyl transferase enzyme. So the gene normally code for sugar that would be recognized as foreign by the human. So the from the pig the organ can be transplanted. The bioprinting, the construction of biological structures by computer aided automatic layer by layer depositing of bio ink onto bio paper. These are the very recent and advanced technology. So bio ink cells from the patient and bio paper are the thin gel layer are used as a bio paper, similar to blotting technique they are using. Then embryonic stem cells. What is embryonic stem cells? The cells derived from a blastocyst. What is a blastocyst? After the fusion of the male gamete and female gamete, we are calling it a zygote, and zygote stage they follows the cleavage. So during the cleavage, the internal cells are cleaved, 2 cell, 4 cell, 6, 8 cell, 16 cell likewise, and they are encircled by the cyst-like structure that is called the blastocyst, and that can be differentiated into any type of cells in the body, because the embryonic stem cells are having that much sufficiency. They are totipotent, pluripotent, they can develop into any type of cells in the body. So the embryonic stem cells are used for treatment nowadays. Culturing the embryonic stem cells in a laboratory. So they are isolating the inner cell mass, then culturing in the medium, having less than 1% proliferation, then they are differentiated into different cells. That is, they can be differentiated into blood cell, neurofibrils, liver cells, smooth muscles, cardiac muscles, etc. So we are taking the blastocyst, and from the blastocyst we are isolating the inner cell mass which is having the stem cells. So adult derived stem cells, cell from the mature tissue that can be cultured and differentiated to become any cell type from the organ of their origin. So the adult derived stem cells means nothing but our bone marrow is having the stem cells, because from the stem cells only the erythrocytes or the white blood cells, platelets, hemoglobin, everything will be developed from the bone marrow. They are said to be the hemopiotic organ, and the production of the erythrocyte is said to be hemopiosis or erythropoiesis. And so the adult derived stem cells, they can be used, they are having the differentiated to become any type of cell from the organ of origin. So hemopiotic stem cell therapy can be doing for various diseases. So problem with the embryonic stem cell therapy, there may be the cure for the tumor formation, because they are the suppressor of the oncogenes. So adult stem cells applications even in heart disease also. The experiment was done in white rats. The heart attack was induced in transferring the mouse, and here they are using the stem cells, and these stem cells will be injected into the heart through the injection, that is 3 to 5 heart after the heart attack, and this is used to cure the heart diseases. Embryonic stem cells, I told you, they are pluripotent, and possible transplant rejection may occur, because after organ transplantation, some of the, because during the organ transplantation what is happening, they are suppressing our immune system, isn't it? Otherwise there will be the high rejection. So after, because of suppression of the immune system, sometime there is side effect or adverse effect may occur. Adult stem cells, they are multipotent, so they are autologous stem cells, and they reduce the chance of rejection. So adult stem cells, because taking from the adult person, they have the power to reduce the chance of rejection. If our body accept, then only, whether it is a kidney or a liver or any part of the body, that can be activated in a patient body. Otherwise, if there is rejection occurs, this may not be suitable for a particular person to have the organ transplantation process. So induced pluripotent stem cells occurs, so mature body cells that have been reprogrammed to change their identities and revert back to an embryo-like state. So through the biotechnology, we can do this process through the regenerative medicine. The induced pluripotent stem cells or the mature body cells have the reprogram. We are reversing the activity of the stem cells. So induced pluripotent stem cells and method using virus to insert the genes necessary for reprogramming. Reprogramming can be done only in the presence of the genes. Induced pluripotent stem cells. What's the application of this IPS? CS means disease modeling, cell and tissue regeneration, then treating the genetic disorder. Because of genetic disorder which cause various syndromes in a person, that is Klinefelter syndrome, Credus-Chart syndrome, Down syndrome, likewise various syndromes occur because of the genetic disorders. So this can be treated by means of the induced pluripotent stem cells. Then therapeutic cloning and reproductive cloning, which also occurs through biotechnology. This is the end of this application of biotechnology. This is highly advanced technology in the field of medicine. Now the doctors and the medicinal related activities are going for the biotechnology for various diseases. Thank you, Ananda.